Alright guys, we're going to start the last video with
Damage to small blood vessels and capillaries
the
frequently
hemostasis
occurs.
animation that's
When these
found on our
vessels are
screen.
damaged,
And
there
here
are
you can
three
see
basic
sticking it
mechanisms
through
that
the
promote
site
hemostasis,
of a
or the
damaged
stoppage of bleeding.
blood vessel. And one of the first things that's going to happen
Following
is the
damage,
blood vessel
there
is
is
going to
an
constrict
immediate
because of the smooth
reflex that
muscle
promotes
surrounding it.
vasoconstriction, thus diminishing blood
And what
loss.
happens immediately are these collagen fibers that we've seen before, but it's been a while, maybe even the tissues unit since we've seen them. Those collagen fibers are going to just sort of fold into this damaged area and create like a little net to start catching platelets.
Exposed collagen
And
from the
once
damaged
those
site
platelets
will
start getting
promote the
caught,
platelets
they're
to
gonna
adhere.
um,
When
start
platelets adhere to the
creating a
damaged
little
vessel,
platelet plug,
they undergo
and that's going to cause
degranulation
some
and release
chemicals
cytoplasmic
to start
granules,
being
which
released
contain serotonin,
to
a vasoconstrictor,
increase
and
the number of
ADP
platelets that
and
join,
thromboxane A2.
and more chemicals, and more platelets, and more chemicals, and more platelets.
The ADP attracts more platelets to
Until
the
we
area, and the
have a
thromboxane
platelet
A2
plug that
promotes
forms, a
platelet
true platelet
aggregation,
plug that forms, and
degranulation, and
this is sort of the
vasoconstriction.
911 response
Thus,
to stop
ADP
as
and
much of the bleeding as
thromboxane
possible
A2
initially.
promote more platelet
But this
adhesion
is
and
not
therefore
going
more
to
ADP and thromboxane. The
allow for
positive
healing that.
feedback promotes
And
the
so there's the
formation of
platelet
a
plug,
platelet
just kind of
plug.
like the Band-Aid.
The final hemostatic
And here's where it
mechanism
gets kind of
is
complicated,
coagulation.
and you'll see
Damaged
I'll
tissue
hear some of the
releases
stuff on
factor
some of the
3,
TV shows that you
which with the
watch
aid of
where
calcium
people having
ions will
surgery
activate factor
are talking
7,
about
thus
more factor
initiating
10
the extrinsic
being
mechanism.
initiated because someone's
Factor
bleeding out.
12 from active
This is all the
platelets
blood
will
clotting,
activate
blood
factor 11,
coagulation
thus
stuff.
initiating the
You don't have to know
intrinsic
the difference
mechanism.
between the extrinsic and intrinsic, but it's just a simple matter of these clotting proteins that are in the plasma that we discussed in the previous video that are going to basically just go through a series of chemical reactions.
Both active factor 7 and active
create
factor 11 will promote
proteins that will
cascade reactions,
cause
eventually
a
activating factor
true
10.
blood clot. And that's where
Active factor
we're at
10,
now.
along with
So all
factor
the stuff that
3,
you actually
factor
play the
5,
video, the narrator for
calcium
this video,
ions, and platelet
I
thromboplastic
might want to
factor,
throw up,
PF3,
kind of,
will
because it's
activate
just a lot of
prothrombin activator.
details that you don't need to know. And so here's the part that you
Prothrombin
should know,
activator converts
is the
prothrombin
simple
to
conversion
thrombin.
of prothrombin to
Thrombin
thrombin, and then
converts
fibrinogen to
fibrinogen to
fibrin.
fibrin.
And that's when we start getting these yellow strands on the screen.
Fibrin initially forms a loose
like
mesh,
sort of like a
but
wire
then
mesh.
factor 13 causes the formation of
More
covalent
platelets to come in,
crosslinks,
some red
which
blood
convert
cells to come
fibrin
in,
to a dense aggregation of
cause those
fibers.
collagen fibers to start
Platelets and
making
red
a
blood
web,
cells become
and
caught in this mesh of
now
fiber,
creating
thus
what's
the
called a true
formation
blood
of a
clot.
blood clot.
And so where this video stops, I'd like to just continue to say that this blood clot is obviously not the permanent solution, but this blood clot would then get dissolved once these tissues are ready to start healing. These epithelial cells start dividing and filling that hole, that gaping hole that's allowing the blood to come out. And then that blood clot will get dissolved or destroyed, because if it doesn't, that's potentially some seriously bad news. And so that's where we're going to take it back to our notes. And so hemostasis, which is that animation that you watched, has kind of 3 phases that we went through. And that is just to stop the bleeding. It does not go through the healing of the tissue, which is also important, obviously. And these are just the details we just went through in notes format. There again is the protein conversion that takes place in the presence of calcium, so that a true blood clot can form. That won't dissolve until it's made to dissolve. It can stay there until the tissue's healed. But again, once those tissues heal, it's important that that blood clot does dissolve. We'll see that in a second. And so here again is just some more vocab. The serum is basically blood plasma without the clotting proteins. That's if you donate plasma, again, that's what the large concern is, or the value is for those clotting proteins for people that need them. And just quick to take you back to Unit 1 of the entire year. This is a positive feedback mechanism, having lots of platelets in an area, having lots of proteins in an area. All the stuff that shouldn't normally be taking place is taking place because of obvious damage, injury, and so more things than usual, positive feedback. And then again, once that blood starts flowing through the blood vessel, it's going to prevent the more clotting because the tissue can eventually heal. Here are some coagulation tests, which are nothing you need to know, but something you may hear of at one point in time in your life. Here is a bruise, basically a blood clot beneath the surface of the tissues. And so in this case, you can see this is a blood clot in the brain, and obviously the brain has hard enough time in that small space that it's allowed to exist, and so anything else in there is potentially life-threatening. Here's a quick quiz. Which of the following is not part of hemostasis? Hopefully a pretty easy one. Multiple choice trick there, just the opposite words basically. Vasodilation is not part of it. It's vasoconstriction or vasospasm, same thing. What causes the platelets initially to stick to the vessel wall? And it is those collagen fibers that make up that kind of outer tube of the blood vessel that start to penetrate inside the tube once the tube has been punctured. And then the chemical needed to form a true blood clot to do all the conversion of proteins in the presence of calcium. Which chemical is last in the coagulation cascade? If you just remember them in the order that they are on the screen, you're good. Alright, so here's the deal with the blood clot, which obviously is abnormal unless it's stopping the blood from coming out, which is then perfectly fine and actually really good. However, an abnormal blood clot that should not be forming is called a thrombus. If that blood clot starts traveling throughout the system, it's called an embolus, and you'll see this in multiple different places, such as the heart, or even the brain, or the lungs. Somebody's going to be presenting or showing us pulmonary embolism on our little blood disorder things. And so something you can do to prevent that is actually just keeping your blood moving, whether that's taking a long trip or sitting for a long period of time. All those, like taking a test for a long period of time. ACT, for example, you might have some flashbacks. That's bad for your circulatory system. Other things that are bad for your circulatory system would be arteries that are clogged or where the flow is slowed down. And atherosclerosis is a culprit where fatty molecules, hydrophobic molecules, get deposited on the inside of the blood vessel, like lipids and cholesterols, and eventually narrow or close that artery, which again, obviously stopping the blood flow, which is super bad news. And then here's the blood type stuff once again, and so if you're comfortable with blood type stuff, you can probably cut the video and move on, but if you want a quick review, here it is. Again, the deal with transfusions is that they're a little more high-tech now, but at one point that was just pretty much like a lucky guess. There's the antigens that are on the outside of a cell and basically triggering an immune response by creating antibodies. And so there's the antibodies that would match up with them, which obviously is bad news because if your blood is clumping, it's tough to make through the bloodstream, and when capillaries are requiring basically single file red blood cells, clumps of blood equals blood stoppage, and again, super bad news. So there's a slide of blood that is clumped, a little bit more accurate than what you saw or will see in the lab, depending on when you watch this video. And here's a good picture that gives you an idea of those ABO antigens and also the RH antigen D that determines whether or not a person is positive or negative blood type. You can see on the outside of those cells just different shaped protein complexes that represent those different antigens, which creates the different blood type. A antigen is blood type A, and so on. So here again are just the two major antigens for determining the ABO blood type. And again, if you have A and B, you have both antigens, and if you have O, like 0, no antigens, and then A or B. Here's an easy rule to remember. Lance Steiner's rule from the scientist that first worked with blood typing. Whatever type blood type you have the opposite antibody. So if you have type A, you also have anti-B. Anti-A inside of type A blood would cause clumping. And so think of the anti-A as like being against the type A. And so hopefully good review here. People with type AB blood, since they have both antigens, they would have no antibodies. And someone with type O blood has no antigens, so they can have both antibodies. And so there's the reason that people with AB blood are universal blood acceptors, recipients, because they have no antibodies that will cause any clumping and can receive any blood type. And this is why type O blood is so popular at blood donation centers, because you can give type O red blood cells to anybody. They have no antigens on the outside, and there's a chart describing the ABO blood groups. Major transfusion reaction is basically like A with B, mixing A with B. Minor transfusion reaction would be like the other proteins involved, the other antigens that nobody ever talks about unless you study the blood. And so there you can see the result. A picture here that's showing, picture showing the potential consequences of mixing blood, the wrong bloods. It can create clumps in a bloodstream and cause a blockage. The RH is talking about whether or not a person is positive or negative blood type, and that is a completely different antigen. So easy to get confused, but also easy to remember if you have it figured out. It's a silly name because of the rhesus monkey, where it was first studied. But again, it's antigen D that you either have if you're positive or don't have if you are RH negative. And so the deal with RH negative people is that you don't make the anti-D unless you've been exposed to antigen D. Again, this is just an immune response. So if you've been exposed to antigen D, your body's going to start creating that antibody, thinking that it needs to defend itself against the antigen D. And so the next time you're introduced to positive blood, you'd be in some trouble because you could potentially have some blood clumping in your system. And so that describes that right there. And the same is true for mothers who are negative blood type and babies who are positive blood type, a fetus inside of them. The first time they have a baby that is positive blood type, the mother will start to create the antibodies upon delivery because the bloods will mix, and now mom has a defense mechanism. If she were to ever have another positive blood type baby, her blood would attack the baby's blood, and the baby would probably not survive. And so mother is given a RhoGAM shot and basically tells her body not to make the antibodies that would start the clumping with the positive blood ever again. So one shot and she's good forever. This was actually the case for my family. So there's a little bit of a close-up. There's what you should see in the lab, little clumps. And here's another good view of that chart that you should get to know and love. And there's a separate page on Haiku for ABO blood type chart that will help you out with that.